Mm-hmm. Hey, hello everybody. We are live. Thank you so much for joining our Longevity Education Hub webinar. We will wait one more minute to give everybody a chance to transition from the waiting room into the actual webinar. And then we will proceed with welcoming our distinguished guests today. We have a really extraordinary lineup. So I hope you are as excited as me to have this webinar today. See, the numbers are still raising, so let's wait until it stabilizes at least a little bit. We have a record-breaking number of signups today, which we will later promote in our social media as well, so stay tuned for the posts. And thank you so much for your interest in continuing your education in longevity medicine. And I see some people are raising hands. Please, if you have any questions, put them in the Q&A box that is available at the bottom of your screen, and we will be answering them probably at the end of the webinar. Okay, and right now we have everybody, so let me officially start this webinar. Thank you so much for joining, and we are delighted to welcome our four exceptional voices working at the frontier of longevity medicine, personalized medicine, and AI-driven health. And let's start with the absolute legend of many fields, and this is Dr. Michael Snyder, Stanford geneticist and one of the leading figures in genomics, multi-omics, personalized medicine, and his work helped define how continuous biological data, which we will be talking about today, from genomics to wearables, can be used to understand health, disease, risk, and finally, aging and longevity. I will proceed with welcoming all of our guests, and then further introductions can be made as we will proceed into the speech keynote part of the webinar. Then welcome Dr. Evelyne Bischof, our wonderful co-founder of the Longevity Education Hub, without whom this community would not exist. So, of course, we are very grateful to the physician scientist and internationally recognized leader in healthy longevity medicine, current president of the Healthy Longevity Medicine Society and medical director of the Sheba Longevity Center. She's helping shape longevity medicine as a rigorous, clinically grounded discipline and is leading the efforts to educate the future generation of physicians in this area. Thank you so much for joining us. Then we have Nika Pintar, the co-founder and CEO of Ani AI, where she works at the intersection of longevity biotechnology, microbiome science, AI, biomarkers, and women's health. Her work focuses on translating microbiome-driven insights into personalized approaches for health span and preventative medicine. And for that, she, of course, uses a lot of continuous health data, which we cover today. And finally, last but not least, we have Bruno Balen, another co-founder of Ani AI, and a technologist working on AI-enabled approaches to longevity, microbiome modulation, and personalized biological intervention for longevity. His work explores how algorithms, diagnostics, and bioactive compounds can be combined to better understand and influence human longevity and health span. Personally, my name is Nika Pintar. I am coordinating Longevity Education Hub, and I will moderate this session today. And without further ado, I will give the floor to our panelists. Please take it ahead.
Thank you so much, Dominika. I think you are way too modest. You are not only the coordinator, you are one of the leading forces of the Longevity Education Hub, and also in the field of cryonics and general biotechnology and AI. We are so grateful to have you and to keep us organized and to make it flow. Today, we are so excited to not only have Professor Mike Snyder with us, which is already a reason enough to, to cheer and to be happy, especially as a clinician, but also the fact that we will present something that to all of us here that we're working as a team on the project is something very dear and near to the heart, which is the so-called ELITE study. The ELITE study that was a part of the semifinals of X-Prize HealthSpan, but much more than that, it was a study that for the first time, I guess, since I have ever seen, was doing an N1 on 31 patients, was doing a deep phenotyping continuously for 8 weeks on a number of patients that were even on interventions divided into 2 arms. So for all of the clinicians out there, you probably will appreciate on how much data can we get during continuous monitoring, and not any data, especially omics. and how we can evaluate them and use them for immediate, okay, today, daily, at least daily change and adjustment of intervention if needed. So I'm very excited, but I should now shush and give the floor to Professor Snyder. Thank you so much for being with us, and please, the floor is yours.
Okay, well, it's a pleasure to be here. I do want to tell you a little bit about, our work and how it relates to deep data profiling, which I think is the basis of today's webinar panel discussion. Excuse me one second. Here we go. Great. So I'll show you what motivates us. It's really this first slide here. Need to get playing from, there we go. All right, hopefully this is all working for everyone. Great. Yep, super. Okay, this is what motivates us. We really practice sick care rather than health care. And the consequence is that we tend to keep people alive, but not necessarily healthy. Meaning people, depends how you want to quantify this, but the difference between health span, the time people are healthy, and their lifespan, the time they're alive, is about 11 to 15 years. Meaning people are living their last decade of life. And I think unhealthy. And we would all agree that we should try to keep people healthy their entire lives. And then, when it's time to go, they would pass away. And then when you think about it, even when we practice healthcare, it's pretty antiquated. People in the US anyway get in a car always at an inconvenient time to show up at the doctor's office. That hasn't really changed that much in the last 40 years. While you're there, they take lots of blood, and from all that blood, and the time you're there, they don't make very many measurements. An important theme of today's, I think, panel is that we tend to treat people, don't tend to, we do treat people based on population averages. And we're trying to change all of these steps. We think healthcare of the future is not going to look anything like this. And especially, we're going to try and keep people healthy rather than alive. And so I think everyone here knows your health accounts for your genome and other factors account for your lifespan. And so you can now measure all of these, some quite well, like we can all get our genome sequence, probably many people on this call have. And then you can also, but we know other things impact your health. In fact, your genome only counts for about 16% of your lifespan. It turns out lifestyle like exercise, food you eat, mental health, and exposures count for the majority. And a lot of this forms fairly early in life, meaning your microbiome and such. So we think it's important that to measure these, and even if you can't measure them directly, which most of us can't, we can measure them indirectly by doing these deep data profiles to see what people's health state looks like. And so this is what we set up. This is what was being alluded to earlier. We set this up a number of years ago where we took a group of people, drew blood, urine, and stool, and then did deep measurements on them. We do sequence their DNA once, but then from their immune cells and from their plasma, if you will, for the part of the blood without the cells, we'll measure many other things, basically as many molecules as possible. We'll measure their modified DNA, we'll measure their RNA, their proteins, that's what these omics mean, their cytokines or immune markers, metabolites, lipids. We do measure their microbiome, which we know is important for health, so gut, nasal, tongue, and skin in particular. We do a lot with wearables. We brought these out when they were just fitness trackers. And this is on top of deep clinical tests, questionnaires, and some advanced tests like stress echocardiograms, glucose control measurements you'll hear about in a second. And so we do very deep data profiling on people. And for this particular study, we did it longitudinally, meaning we would sample them every 3 months while they're healthy. And we've been running the study now for 13 years. Actually, one of the participants we've been doing on me over 16 years. And so we do take extra samples, I forgot to say, when people get ill. And the idea is to really understand what a healthy profile looks like. How's it change over time? What's the differences between people? And this is an important theme. We're all different. And so the difference in my biochemical profile when I'm healthy versus when I'm sick is smaller than the difference between me and the listeners here. We're all very different, and that's a very important concept. And because of that, as we were tracking these people, we made many major health discoveries. So it was a small group of people. We started with 109, but we wound up making 49 major health discoveries. So hem-oncology, cardiovascular disease, infection, metabolic disease. And some of these were all found, I should say, with before people had symptoms. So and some of them were a big deal, like we caught some with early lymphoma, two people with pre-cancers. One has recently turned into aggressive cancer, which we caught because, again, we were tracking them closely. We did find two people with serious heart issues, one from genome sequencing, one from the wearables. And so the important lesson from all this is that no one technology found these. In fact, it was the SPECT suite of technologies that uncovered their health profiles, if you will. So we're getting a much clearer picture of people's health. We like to say we're getting maybe 700 or 800 pieces out of 1,000 piece jigsaw puzzle versus 5 or 6 we would argue medicine does today. And the longitudinal profiling is very important. We could also see, because we were tracking healthy people over time, how they change, and it turns out that everyone is changing differently. So in fact, this person here, their top biochemical pathway is cardiac hypertrophic signaling pathway. We later learned they're stage 2 hypertensive, so this sort of made sense. This person had their immune and metabolic pathways shifting over time. And so we call these aging patterns ageotypes. And once again, everybody has a different pattern, meaning if these are the 4 ageotypes we uncovered initially, and each column's a different person. These people age in all 4 categories. This one, not much of a kidney ager. This one, kidney, not much else, kidney and metabolic, and so on. So again, we're all different. And you can pick this up from these deep profiles. And it turns out there's actionable information. There are clinical markers associated with these ageotypes, and people actually acted on that and improved actually their markers. We're also very keen on remote monitoring and wearables. So if you're looking up, I'm wearing 4 watches and 2 rings. Even my hearing aids are sensors, and they measure pretty important things: heart rate variability, blood oxygen, many other measurements you can make. And so we discovered early on that when we put these on a cohort, you can tell when people are getting ill. In fact, we could first tell when I got my Lyme disease from a simple smartwatch and a pulse ox because my heart rate went up and my blood oxygen went down. And we later went on to show you can tell when people are getting respiratory viral infections from this. So when and we published this in 2017, and COVID came, and we wrote a real-time detection system. This is definitely going to be the future of medicine, where you're tracking people and you can see shifts from their baseline. And so for the case of COVID, we can see shifts from people's baseline in advance of symptoms. And the median is 3 days. And it works. We have a real-time detection system that works 80% of the time, meaning we will see red alerts, if you will, before symptoms appear. And then they get diagnosed typically the next day or the same day these days. So it's a very sensitive assay. And again, digital phenotyping, we think, is going to be the future of tracking people. I will say these continuous glucose monitors, if you want an illustration of how important personalized tracking is, these continuous glucose monitors measure people every 5 minutes. And given that we have a diabetes endemic that's worse than the COVID pandemic, we think these are going to be important technologies, meaning in the US, 11.6% of people are diabetic, 20% don't know it. 38% of people are pre-diabetic, 80% don't know it, yet most will become diabetic. And so we discovered early on that when you put these on so-called normal people, some do have good glucose control. Many are moderate spikers, but some are severe spikers, just as bad as diabetics, meaning their glucose is going very high and they had no idea. And then we also quickly discovered, as did Aaron Siegel, that different people spike the different foods. So most people will spike the white rice that's shown here. And for most people, this is worse than ice cream. Some people are pasta spikers, that's green, potato spikers in yellow, bread in blue, or grape spikers. We're all different, very personalized response to foods. And we think the basis of this is the heterogeneity of glucose dysregulation. So glucose dysregulation involves many organ systems, your pancreas, your liver, your muscle, your intestine, even your brain's a big glucose consumer. many biochemical signaling pathways, insulin, glucagon, GLPs, I'm sure many of you are familiar with, all play a role in glucose regulation. So we went in and subtyped people, and we could show that everybody's different when we subtyped them from muscle. Here's their hemoglobin A1C. So the blue is normal. These are pre-diabetics, the diabetic. So basically, what we discovered is that when you subtype people for muscle-resistant beta cell defects, incretin, hepatic, you can see the pattern. Everybody's different. And just to give you an example here, this person's insulin resistant, but normal for incretins. This person has a severe incretin defect, but normal for muscle resistant. And with machine learning and AI, we can actually just get the shape of a glucose curve and deduce people's subphenotypes, at least for muscle resistant beta cell. Again, so this can now be a very simple test. Why do you want to know that? Well, determining your subphenotype determines what food you'll spike to. So if you're muscle resistant, you'll be a potato spiker or a pasta spiker. But not if you're insulin sensitive, then you don't spike to those. And not only that, I won't show you, but you can mitigate these spikes by eating either protein or fat ahead of time. But that depends on your subphenotype too, whether that's successful or not. So again, we're all different. We can even take this further. We can actually subtype people, track their activity. Once again, in the era of machine learning and AI, you can make very personalized predictions about people's response to their glucose, if you will, levels and basis of what they do as well as when they do it. And some of this we discovered is already known. If you eat your big meal first thing in the morning, you tend to have lower glucose late at night, not so good. Higher glucose, starchy vegetables, bad. Fruits, good. Most people don't sleep enough. Sleep more, you can lower this. This is also a big deal because for muscle insulin resistance, if you exercise more, you will actually improve your muscle insulin resistance, but not if you're a beta cell defect. And it turns out I'm a type 2 diabetic. I'm a beta cell defect. I gained 10 pounds of muscle mass to try and control my glucose. It failed because beta cell defect doesn't matter how much muscle I have. So your subphenotype we think is very important. And again, emphasizing the power of personalized medicine. I'll just wrap this up quickly by saying that we do a lot of remote monitoring where you give little samples of blood. We think this is the future. You overnight them to the lab. This complements digital monitoring as very well as well. And then you mail them to the lab. And these days, we can make 7,000 measurements from these little drops of blood by doing these deep omics assays you were just hearing about. And then we discovered everybody reacts differently to different foods. So this is just showing 3 simple carbohydrates. Each box is a different person. This person's carbs goes down when they drink the shake. These go up, up, up. This one's flat. That goes down. That goes down. We can classify people once again into groups. So the gray is the average of all 32 people who were in the study. These are inflammatory markers, and brown for this group of people, they go down when they drink the shake. This group of people goes up. Same shake, pro-inflammatory on some, anti-inflammatory on the others. So bottom line is you can see we all have very, very personalized response. So I'll just wrap up because I want to save time for a panel discussion here, but to say that we think the future medicine is in fact tracking deep data longitudinally. We think digital markers is really the future of medicine with some sporadic home sampling to actually collect additional data, biochemical data. And together with all this and your own AI agents, we'll be able to better keep people healthy, happy, and hopefully living long lives.
Well, that's the future I definitely want to leave at. And so thank you so much for this. I personally jotted down 7 questions and my personal inbox is overflowing with questions to you, but let's leave them a second for the later part of the webinar to have the 4 perspectives combined. So who dares to go next after Professor Snyder?
Nika, would you like to share the screen for our slides? It would be really great. And I can start by a short introduction, right? I don't want to take too much of the stage because actually Nika and Bruno are really the engine behind what we were doing. But just to mention maybe once again the context, right? To put it down. We were trying, first of all, to have a tool where a clinician can have a very clear and very good possibility to track and monitor a person digitally and with markers. And some of the goals was also to really make sure to compare those together. I understand that my camera is playing crazy. Please forgive me for that. But sharing the screen is the most important, as long as you can hear me. Great. So we really wanted to make sure that we can take a person, very few, very sparse data of the person, and take it to the next level and get as much of a complete biology as possible. And that was the study that we did. Maybe next slide. The study was called ELITE. It was conducted at the Shiba Medical Center in the Shiba Longevity Department. And you are all very familiar with the Healthspan X Prize, so I will not mention that. We actually had Dr. Jamie Justice speaking here, so that is what was happening. I just might want to mention that any AI is both semi-finalists in the X Prize Healthspan and finalists in the additional prize of the X Prize Healthspan, so it's pretty impressive. Now, really, from a clinician to clinicians, what did we do? Here you can see the study design, and it's not like the typical study designs that you might have seen in all of our careers, where we have usually 2-3 groups randomized and crossover. Here we see several very exceptional points. Number one is it was a study conducted within 8 weeks. That was also the concept of the X Prize Healthspan. It has to be a quick intervention with good result. So quick, cheap, and then also scalable is basically what is the concept of X Prize Healthspan here. We were lucky to recruit 31 participants who were then divided into 2 groups, both who took both supplements, a specific mix of supplements, minerals, vitamins, and prebiotic, and repurposed drugs. Specifically, we chose metformin as a base for both groups and added either a semaglutide, it was specifically Ozempic, or an SGLT2 inhibitor. In our case, it was dapagliflozin. We used the minimum dose of each of those medications, the minimum, not microdosing, minimum dose. Just to mention, that protocol had to go through ethical committee, a very, very strict ethical committee. So one of the fun part was actually to demonstrate, and thankfully you have the data now, that repurposed drugs, as we see here, actually do act on pathways of aging, and did show some of the aging reversals in several crocs. And of course, they were very safe. Now, what happened to the patients? They came to us several times to the hospital and received very comprehensive assessment on day 0 and after 4 weeks. And they had also a midterm semi-assessment, which was extremely comprehensive and included, first of all, stamina with all the body compositions. It included cardiovascular path, included a CPET with the VO2 max and all of the cardiovascular main ports, arterial stiffness, and of course, all of the biomarkers that were associated with those domains. Sleep was a very important part for us, and not just any sleep, not with a personal wearable only, but actually with a sensor, where we were able to identify sleep apnea and a very deep level of sleep architecture. The same goes for HRV measurement, and then a full metabolic panel in the hospital with the blood, with the standard way of taking the blood and measuring all the markers, followed by a very extensive station of brain health, cognition. And we will talk about it a little bit later, how deep it was. Now, on top of all of this, we also looked at specific parts of the blood tests, focusing on menopause and andropause. Our patients or participants in the study were 50 plus. And then came the magic. We also did sampling for the full omics, which Nika will talk about in a moment. And also we used digital and multispectral device to track the patient daily. So it was a group of patients who were very heavily loaded with daily monitoring, with daily testing, with daily pictures that they had to take from their tongue, from their eyes, from the bone, so the wrist, from the cheek. And there were also several questions and even cognitive testings every single day that were associated. That was the design in a nutshell. It actually went much deeper. But let's go to the next slide. Point in case for a clinician, it's a group of patients who are 50 plus, so not teenagers, and very, very heavily loaded digitally. Why? Because we really wanted to have an algorithmic polygenic phenotyping of the patients. It's really a case study. It was a pilot. It was supposed to be the pilot for the actual bigger study. But what we saw immediately and what was very reassuring was, number one, that patients had an exceptional compliance. Exceptional compliance in a set where you have to take medications and supplements and liquid, and you have to test yourself every day and to scan, might usually end in really compliance fatigue and dropouts. None of this happened. In terms of results, we had only responders and super responders. We had 2 patients that were non-responders, probably because of specific genetic variants that made them very sensitive to metformin, believe it or not. And we had no serious side effects. We had no serious side effects for all of those patients all along this time. Next one. So what did we see all of that along this case, along this time, besides of the fact that patients were extremely motivated along all 8 weeks during our time? Well, first of all, we saw the main result, the main improvement in the cognition. So X-Prize is working on several points of immune, brain, stamina, muscle, but in the cognitive, we checked cognitively several domains of cognition. This was including a general cognitive state. It was including attention, verbal memory, and executive function. And actually, in all of those 3 domains, we've seen an increase and improvement. And this without an actual brain training every single day, which would have been an intervention. So this is all based on the fact that the patients were actually involved and engaged. Next one. What was very interesting for us doctors, of course, was the metabolic change. And we indeed saw a lot of changes here. We are listing only those that were very prominent and very clearly statistically significant, but there were more and there were a lot of trends. But what was very interesting, of course, with repurposed drugs that work on diabetes, you will see a drop on fasting glucose and general risk of prediabetes. That was expected, yet we were very happy because it's not always the case in people who are 50 plus, who had absolutely no change requested from our side in terms of their diet and nutrition. They were asked to continue with the same baseline with everything what they did. And they actually reported that they kept the same diet. Also, their cardiovascular score improved, NT-proBNP, one of the most sensitive markers, I would say, for that has dropped. So we did not use any medication for uric acid control. The increase of water intake was not a case in point for sure, because we also measured other kidney function points. So we saw uric acid going down, and it was very beneficial for a lot of our patients. And with the supplementation, we also had an increase in vitamin D level that was very beneficial for those as well. Great, next one. So here I think Nika will already jump in. Yep, Nika will be jumping in now to the next.
Great, thank you, Evelyne. It's a great honor to speak right after you and Mike. What I would like to emphasize here in ELITE was also that in the 8 weeks, besides the cognition, which we were looking at the multiple domains in the Sheba Clinical Center, we were also doing validated questionnaires. And what we've also seen was lower anxiety and depression. What was quite interesting was also the study was conducted in Israel during a very interesting geopolitical situation. This was not expected given the circumstances. However, we have also done an additional level of cognitive testing through our ANI app. which has also confirmed in other cognitive quizzes, such as that were more about reaction time and so on, that also cognition improves. So basically, all the different domains of cognition and brain health that we looked at, we've seen improvement that was statistically significant, which was beautiful. But not only that, we went further and did various omics. We are not here now presenting all of them. These are all preliminary results. Please keep in mind that we basically wrote the protocol in July. In October, we started recruiting. In February, we finished recruiting, and all the data that we have here is literally from mid-February until today, which is very little time in clinical practice to just even analyze all the vast amount of data that we got. But what was very interesting was also the results we've seen in the oral microbiome, where basically we had strong significance when we were looking at the oral species richness, which almost doubled, both in intention to treat and per protocol set that we've done. But not only that, we have seen in other domains, we have seen weight reduction, fat mass index went down, BMI went down, but also in DEXA scan, actually, we've seen that when it comes to muscle health, we did not, what was expected that people would be losing muscle, but actually other domains were also positive. So all of these favorable results, what is very important is that we've seen multi-systemic effects. So it was not just one system, rather various systems. And keeping in mind, this was only 2 months without any lifestyle interventions. So we intentionally, in this protocol, designed it in such a way that we had supplements, we had repurposed drugs, but no lifestyle guidance, knowing that basically lifestyle, we know that it influences longevity beneficially. So what we wanted to see here was, what do molecular interventions do, and how can we then, for the final, structure the design to also include lifestyle interventions, which we believe will further influence the results beneficially. What we've also done is responders analysis. So we went deeper into the participants that had a greater response. We were looking at multi-domain response. And besides cognition, we have also done glycan analysis. We've done transcriptomics. We have epigenomics currently being analyzed. What we've seen, for example, in glycan biology, we had a responder that had within 2 months a 10-year reduction in glycan age, which was something that no one expected, not even the glycan lab, again, it's from Croatia. Because it was only a 2-month intervention. Usually it takes a longer period of time to even see any movement in glycans. As Evelyne already went through the metabolic markers, we have also seen in transcriptomics some very interesting immune changes. What was very interesting there was that while we've seen the change was more related towards a beneficial immune fitness, as in the whole group, we didn't have a CRP level going up, so it went down actually, the overall trend. We did not reach statistical significance, but the overall trend was down, which was very interesting. And specifically in the responder group, this effect has been the most pronounced. In some RNA age that we calculated, it even went down 4 years. Of course, we cannot look at various clocks in isolation. We need to look at the whole picture. And that's why we are doing this profound analysis of responders and non-responders to understand how we are shifting them. And now the main point why we are doing all of this is that for the finals, we can actually look at how can we place responders into the super responders category, meaning that they respond across multiple domains, specifically immune, cognitive, and muscle function. And how do we move the non-responders into the response terrain? And the whole, one of the underlying concepts that Evelyne touched upon was the point of daily imaging through our ANI app, as well as voice scanning, getting context about one's lifestyle, was to reach the point where we can earlier stratify who is the responder, who is the non-responder, and who is a super responder, in order to have as much as possible real time early stratification to understand whose medication should we continue, whose intervention set should we continue, whose should we change, which parts of it should we adapt, and what should we actually stop? Because one of the questions that we are rarely asking is actually, what if something is just not beneficial for a certain person? Whereas, um, even if we would, based on existing literature, think it may be beneficial, perhaps for this individual, it's not the right solution. And therefore, it's really important to understand as soon as possible, what do we need to change and when. So we built the ADAPT engine based on Bayesian modeling, which is also in line with the most recent FDA guidelines for using Bayesian methodology in clinical trials, as well as implementation of real-time clinical trials. I also wanted to include these slides just to give a broader overview into the clinical practice and where we are heading, as well as an industry, which is extremely important. And basically, the whole ANI platform has already been built for that. The digital platform is already in use in multiple universities and research centers. We are running multiple clinical trials. There is one which is very interesting, called MetaboTwin, that we are doing in the Netherlands, where we are actually having a very specific set of dietary guidelines. You're monitoring what people are taking and also doing multi-omics and digital profiling, which is similar to what Mike was talking about, to really see also the glucose response across and also other multi-omics domains. So we are feeding the system. We are doing the longitudinal monitoring. So participants are doing the daily scans. They are taking various images from their eyes, face, and so on. And what's even more important, there is an interface to discuss through our Talk to Ani feature, so that they can basically provide additional contextual data about one's daily life. On the other side is the physician support system that was also utilized in Elite, where physicians can actually monitor what is going on, what is the calendar, when are the participants coming, what should they do, and so on. I won't dive too deep into this, but just a couple of images what it looks like. And I am now giving the ball to Bruno.
Thank you. So in order to make aging biology, to understand it deeply, we cannot look at it through one biomarker. We know that aging is progressive loss of coordination between biological systems. Next one, please. So we made a framework. We will actually publish it, Mike, Eva, Nika, and I. We call it a coherence framework. It's actually really simple. In the middle, it's theoretical, perfect coherence, super coherence, where everything is perfect coordination. It's only hypothetical until now. On the edges of the circle, it's dead. So that means that we are losing coherence as we are aging or becoming disease. Once the pattern of decoherence becomes visible and observable, we call it a disease. That means before that, we still have that pattern happening, but we cannot observe it yet. People who are educated to observe this pattern, they're very well educated, we call them physicians or doctors. And what they do when they observe that pattern, they match it to intervention that is restoring that particular pattern, and we call that pharmaceutical. So in that sense, if you look at it from first principles, pharmaceuticals are not made, and current medicine is not made to restore communication across systems. That means it doesn't target system itself. It targets downstream biomarkers. It can be anything that can be traditional target, or it can be aging clock, but it's targeting one thing. What we are working on is to understand how all the systems move together, how they communicate, and how we can actually capture the pattern itself. We also published one paper that is creating a structured coordinate system of biology and delivering it whether to AI agents or physicians as a structured object that is deterministic in sense that is preventing LLM vibing about one's biology, because it's giving the position of coherence within this coordinate system. More practically, next one, please. The question is, how do we measure the system itself? It sounds really abstract. And if you look at this person with a kite on a beach, and now if you would hide the person and ask you where is this person standing, just based on the kite, it would be really hard to approximate. We don't know the length of the rope, we don't know the wind, we don't know the angle. But that is what we are trying to do today, to see one biomarker and to reconstruct this whole space, to reconstruct the aging biology itself. Next one, please. But if we would give a dozen or a couple of dozens of biomarkers, and that is reducing potential surface area where one could be standing. So that means that we are still not seeing the pattern, the system itself, which is impossible, but we are reducing the error that we can see how all these systems move together. Because if now the person would be moving, all these kites would also be moving, that we could still approximate this lower surface area. If each of these is presenting one biomarker, one aging clock, one endpoint, cognitive domain, even picture or HRV or something that we are measuring, then we can reconstruct the system. And the problem there is it's really expensive to do daily multi-omics and functional tests and cognitive tests, cognitive batteries, and it wouldn't be feasible. It would take a lot of time. It would be very expensive. It would be a huge burden. But it is the right way. So how we are approaching this is, next one, please, is that we are collecting all of this, all of the data. We collected all the omics in this one. So from epigenome transcriptomics, we did proteomics, we did null-ISA immunomics, microbiome of stool and saliva, 20 million readings. We really went deep. Now we are waiting for epigenome and mitochondrial genome. We did comprehensive blood work with all the hormones. We did all the functional measurements and cognitive batteries and daily digital phenotyping, because we want to compress all of them in one latent space and see if they're landing in the similar position. If they're landing in a similar position, then we can do ablation and we can cut certain biomarkers, we can remove them, and we can still see and approximate similar position, at least similar or trajectory within that state space. That means that we believe that future of diagnostics is digital, really non-invasive, and it's sparse. But in order to reconstruct the whole thing with sparse input, the whole system, and to update it daily, because that is basis of in silico trials, to have daily updating of this world model of in silico of a digital twin, not have it as a snapshot. We must have all of them together. When we have once all of them, then we can remove them. Then we can see how precise we are in that. Because how would you cross-validate something if you didn't have all of them in the first place? So what we also want to make is this positive pressure on the field, and Mike's lab started it. It's to 2,000-fold the data yield per patient. We should maybe million-fold the data yield per patient, because all the data that we were taking until now was optimized for human interpretation. And human interpretation can see maybe a couple of biomarkers, but not complex relationships between them. What we have today and what we are about to have with all the super intelligence that is happening and processing and speed power and multi-omics and digital, we can just increase it, and we can create a biobank as a field that will be enough so that we can reconstruct the biology fully. So this is our approach. It's also looking at, we call something healthy aging, but healthy aging is still a loss of function. We still have pattern of decoherence. So what aging clocks are doing, which are really valuable, and we did all of them, is that they're looking compared to a reference group and population, but not compared to your supercoherence. So you're being compared to people your age, which are already decoherent. So that means if we see 50 age younger, biological age, that doesn't mean that you're actually 20 in all the functions. So can we look at this local coherence, meaning comparing ourselves to our chronological age, but also working on the restoration of coordination between systems? Because if we restore the coordination, then the system itself is moving toward more coherence. And it is the theoretical framework, but we believe that is solving the problem of how we measure aging and how over time we will be doing it real time while we don't have any visible signs of diseases. So whole elite was done to prove that this is possible, and we are seeing great results. We can stratify responders and non-responders with 86% accuracy only through digital already. We see dozens of markers with more than chance as they are moving together only from digital. We still don't have it on the level of sensitivity and specificity that we go to have and offer it as a full clinical test. But as they're all moving together, we can approximate trajectory, direction, and position in state space. So this is our approach. We will continue much deeper, much more. we learned a lot, and this is we are still just touching the surface. What we can measure today and how often we can measure it, it's we can do really something special. We can reconstruct our biology and then not only prevent diseases, not only prolong our health span and lifespan, but also enhance our cognitive capabilities. How do we feel? Because these people weren't depressed and anxious, and they had reduction in depression and anxiety. They weren't cognitively impaired. They have amazing results in cognitive function. They didn't have sarcopenia. Their muscle function improved. So yeah, this is part of our work. We don't have to go into all the measurements, but we will be publishing many papers through this. We, for example, saw that many aging clocks are orthogonal. They're showing exact opposite change. That doesn't mean that one clock is better or not. Like Mike always says, the best clock is the one that gives you lowest age, but it just proving that the biological change is real. So maybe we can stop looking at it as a scalar, as a one number, as one clock, and observe it as aging map, as a map of interactions that is making this pattern of our biology. And last but not least, we believe that since aging is a multisystemic problem, it is happening in dynamic nonlinear system, that is human body, that it must be targeted through polytherapy. It must be adaptive. It must be something that is adapting to this nonlinear system. And that means that it cannot be treated with one molecule or one intervention. It can always be combination that is curated for N of one. And that is the only way how we are not actually leaving anyone behind. We are not leaving anyone behind, because if somebody is non-responder, we are not saying, okay, population average is still good, let's put a drug on the market and not care. We say, okay, what can we do for this person? And what is the next one? And what is the fastest way to see whether they're responding or not?
And on the note of not leaving anybody behind, please allow me to say first and foremost, thank you to all of the panelists and Nika and Bruno for having us involved in this great projects actually, and then from my side, first of all, a big thank you to the entire team at Shiba Longevity, Professor Strauss, Professor Goshen, every single person, therapists, nurses, doctors, everybody who helped, and of course a big thank you to patients. There is actually a lot of them in the audience, and they have been very much looking forward to seeing the first glimpse of their results. They will all get their feedback later as well. They're all asking when it's continuing. So thank you for holding up with us, for being so active and making healthy longevity medicine progressing faster for the world. Dominika, thank you. The floor is yours.
Thank you so much. And now it's my turn to thank everybody here, Professor Snyder, Professor Bischof, Nika, Bruno. Thank you for your time and for this great talks about continuous data monitoring and how the future of our health will look like. And I have done a lot of sorting through questions here, and I ranked the most interesting for the audience. So let me just throw them to you, and please, whoever is the most interested in that specific one, please answer. The first one is, how do digital values relate to actual values? And what is the sensitivity of digital phenotyping compared to the actual one? Yes, please.
Yes, so we still don't have, as I mentioned, the significance that would be specificity and sensitivity to become clinical, to replace clinical biomarkers. But what we see is a trajectory, and this trajectory of movement across systems is something that is quite accurate. So that means that I cannot say exactly this protein changed and this bacteria is now prevalent, because we are nonlinear systems. So we cannot yet find it on this N, on this number of people. However, what we are seeing is that we are seeing dozens of different markers beyond chance that are moving together and that are following this response versus non-response. Because if we can track response and non-response in real time, then we can have adaptive polytherapy. I also must say that approximately 2/3 of the targets that we can see weren't visible only through phone measuring, but our multispectral device that we have developed restored them. So it helps restoring them by increasing the surface area that we are giving to AI to find correlations.
Okay, thank you so much. I hope the person who asked the question is satisfied with the answer. And we have the next one directed specifically to Professor Snyder. So somebody pointed out that they were very shocked that you are wearing 4 watches and 2 rings. So I guess it's for the comparison purposes. But if you have so big insights into both the academia level of data analysis and the consumer facing level of how it's actually measured, what do you think will be the next big thing available to patients? Is it the platform that will allow for continuous monitoring, or is the specific device? What's the next thing to anticipate?
Well, it'll probably be both. We definitely need a platform that integrates all the different forms of data, the types of data that Bruno talked about, from facial scans and other things, and as well as the smartwatch and other data as well. So definitely having platforms come in that integrate this data and give feedback to the user. We built that for COVID detection, and we're building it for mental health detection, like depression and anxiety. But it doesn't exist for most things. I can give you a specific example in our lab where someone actually died after a strong exercise. And yet, when we went back and looked, his wife shared his data from a smartwatch. And it was very clear 4 months before he died, there was a big step function and change, meaning his heart rate variability dropped, his heart rate went up, gait changed, a whole series of things, some of which Evelyne had talked about earlier. And the bottom line is the shift was there, but there was no mechanism to relay it back. There was no platform there. So we definitely need the platform. But I do think these devices will continue to evolve and change. Glucose monitoring is quite powerful, and a lot of creative ideas out there for how to do that off a watch rather than off of these patches. So both technologies will push their way forward.
Right, definitely. And I think especially for the long-term monitoring is important because the current version of the Apple Watch or the Samsung Watch just gives you, for example, for sleep, the last night score. And of course, it gives you the patterns. But for a week, I don't think for anybody who is not that into medicine, it means anything specific. So we need somebody higher, like a medical big brother, to be looking over that.
Yeah.
Okay, the next question directed to Dr. Evelyne Bischof. For clinicians specifically, from a physician perspective, how does the concept of digital data monitoring fit to choosing wisely?
Thank you for this question. Whoever colleague is in the room, choosing wisely is a very core concept in medicine that was developed, I would say, about 10, 15 years ago. It started with smarter medicine, then in choosing wisely by saying it's not only about we prescribe or we deprescribe. It's all the continuum on where we do smarter medicine by less is more and where we actually add things where it's necessary. So the concept of choosing wisely is tapping on pre-AI era, pre-phenotyping era, on the fact that we as doctors, we really want to individualize ongoingly. We want to have a continuum and we want to have a support system to our, human coherent circle that we are running in our heads as clinicians, that is much better than us and can hold up with our hundreds and thousands of patients if you are in a hospital, or or more if you do it over the years and you actually track longitudinally. So it fits right there, and the adapt engine that Nika mentioned is doing exactly that, right? Every day you have a checkpoint and then you see, do I go up, do I go down, do I don't change? Not changing and keeping the optimum is a hard work as well. So yeah, sounds familiar. It's just now a little bit more automated.
Right. Just like Bruno mentioned, now we have a smarter colleague, the AI, that will be able to process much more data, so we can also spree a little bit on what we measure. Okay, we have another question for Nika here. In Ani AI, can others access your data from the research? And if yes, in what data standard are you using?
So the data that we are collecting is our proprietary data that we are sharing with researchers depending on the specific project. Of course, if someone is interested in collaborating, please reach out to us and we can see what would be the possibilities to collaborate.
Great. When we will be sharing the recording of this webinar, I will also put all the company information and contacts.
If I may add, is that the biobank itself, meaning all the data, is proprietary, but the algorithms that are coming from it and the ability to run inference from all these connections and using the application to early stratify responders versus non-responders is available. So while the data is available for researchers directly, for everyone commercially, it's available to run inference on these models.
Oh, that's very useful to know. Actually, we'll also highlight that. Then we have another question that that accounts both for what Dr. Snyder was talking about and for the part of the ELITE trial. So the question is, how do you account for diet and combination of foods such as rice and fiber when defining metabolic subtypes?
Yeah, I'll start. In our case, we start with people fasted to make that subtyping I mentioned. So I didn't get into details there, but like they'll drink a shot of glucose while fasted, and we'll look at the shape of the curve. And with machine learning and AI, now deep learning, we can tell what their subtype is. So they're generally fasted, and then for actually measuring the true subtypes, we use the state-of-the-art tests. There's something called clamp tests and OGTT. The physicians will understand these. And that lets us precisely tell the subtypes. But these days, we can do it just from the shape of a glucose curve, and they're fasted to begin with. And the curves are fairly reproducible as long as they're fasted.
Okay, thank you so much. And then finally, we have the final question, because our time is coming close to the end, is how do you see the use of continuous data as clinical evidence and endpoints for clinical trials and for drug regulators? Also a very broad question.
Well, I can start with that one, but I think others should weigh in too. I think in the end, in the US, you'll have to get FDA approval for these sorts of things, and they will become a new form of diagnostics. For the wellness case, which is how these things usually roll out, you can bring them in and make lifestyle recommendations, things like that. But as a true clinical test, you ultimately have to get the algorithms approved, and there's a whole FDA mechanism for that.
Okay, I'm sure it will be very helpful for any of our listeners who are planning to do their own study. Thank you so much to all our panelists.
Dominika, may I add something regarding this one? I think that it's really important that already today we can have physician supervised adaptive polytherapy, meaning that physician is making final decision, and I think it should stay like that. But physicians should just be better informed with AI. So instead of replacing them, giving them superpowers. And I think that we're really close to that, and digital will increase the frequency, because if we can have these black boxes in middle of visits, between visits, much more visible, and then true frequency. If we see any drift that is maybe not as deep and as with high resolution, then we can do omics. And often ask, is future digital or omics? Probably in the future with digital, we'll getting more data, or equally as we are today with omics. But omics will also go deeper. So that means that we'll have frequency and resolution when needed, and we will be combining those not because we are feeling that it's time for our annual exam, but when something drifts and when a physician is being notified that they should do something.
Yeah, that's a really good point. If I could add to that, you can actually bring in the concierge services, for example, in the US, bring in wearable device and digital data into their clinic to make recommendations to patients.
Yeah, and I would add what's really important is this part of the collaboration between the innovators and the institutions and the regulatory frameworks, which is precisely why I emphasize these new guidelines by FDA, which are very positive and looking towards a future that is truly N-of-one, and where personalized medicine is not just another thing that we are discussing, at the aging biology conferences, but rather it is a proper framework And therefore, if we have a framework, we can move it forward. And I would like to thank also Shiba Medical Center for recognizing this, because we went through all of that when we were going through the regulatory approvals and the ethical approval for the ELITE trial. And, you always have to put in so much work to get all the paperwork done and everything in the right place, and that the infrastructure also follows that. It was quite a big milestone for us as well. And I would just like to thank the team also for being so open to bring this innovative concept into the clinic and ultimately to the patients, which matters the most.
Thank you so much, everybody. And I really like how we scooped this last question, because it allows me to piggyback on the major trend is that physicians are facing a huge change into how the profession is being executed. And that's why Longevity Education Hub was founded, and that's why it exists. So with that, I would like to thank you very much to Dr. Snyder, Dr. Bischof, Nika, and Bruno for your time and for your wonderful, I would say one of the first presentations when it comes to ELITE trials specifically here to the general public. So thank you so much for your time and for your trust in our audience. And with that, for all our listeners, I would like to invite you to rewatch the recording of that to get all the nitty-gritty of the details, as well as to take our courses on the longevity.degree website, available in 8 languages, that will prepare you exactly for the entry level of what was discussed today at the webinar. Thank you very much for your attention.
Dominika, if I may thank you,
Please.
because what you're doing, if you remember, the coherence circle is this part that was invisible until now, and that is what how do we treat somebody without seeing pattern of disease. So I think it's extremely important. It's future. It's needed to fulfill the whole circle.
Exactly. We need to fulfill the circles.
Yeah.
Thank you so much,
you.
everybody.
Thank you so much, Dominika, for great moderation. Really appreciate it.
Thank you. I
really appreciate
the level of our speakers. Thank you so much. We will see you in the recording. Thank you.